Early anatomical studies, 1543. In 1543, the publication of Andreas Vesalius De Humani Corporis Fabrica represented a monumental leap in anatomical science. This comprehensive work challenged the long-standing anatomical theories of Galen, which had been accepted for over a millennium. Vesalius' meticulous dissections and detailed illustrations provided a more accurate and complete depiction of human anatomy, particularly the nervous system. Vesalius emphasized the importance of direct observation and dissection, moving away from reliance on animal dissection and second-hand accounts. His work included the first detailed description of the structure of the brain, the meninges, and the cranial nerves. By providing accurate representations of the brain's anatomy, Vesalius laid the groundwork for future explorations into the function of different brain regions and their roles in human behavior and cognition. Discovery of electricity in nervous system, 1791. The realization that electricity plays a fundamental role in nerve function came from Luigi Galvani's groundbreaking experiments in the late 18th century. In 1791, Galvani discovered that electrical stimulation of a frog's leg muscles caused them to contract, even when the muscle was detached from the brain. He proposed the existence of animal electricity, suggesting that electrical forces were integral to muscular and neural function. Galvani's discovery opened the door to electrophysiology, the study of the electrical properties of biological cells and tissues. His work laid the foundation for understanding the bioelectrical nature of neural communication, which would eventually lead to the development of technologies such as electroencephalography, EEG, and the therapeutic use of electrical stimulation in conditions like epilepsy and Parkinson's disease. Localization of brain function, 1861. In 1861, the French physician Paul Broca made a significant discovery that would shape the field of neuropsychology. Broca studied patients with speech impairments and found that damage to a specific area of the left frontal lobe, now known as Broca's area, was associated with the inability to produce coherent speech. This finding provided strong evidence for the theory of localization of function, which posits that specific areas of the brain are responsible for particular functions. Broca's work was crucial in shifting the scientific consensus toward the idea that the brain is not a homogeneous organ but rather a collection of specialized regions with distinct functions. This concept has been fundamental in the development of brain-computer interfaces, BCIs, and other neurotechnologies that rely on understanding and harnessing the functions of different brain regions. Invention of the electroencephalogram, EEG, 1924. The development of the electroencephalogram, EEG, by Hans Berger in 1924 was a revolutionary advancement in the study of brain activity. EEG involves placing electrodes on the scalp to record electrical activity generated by the brain. This non-invasive method allowed for the observation of brain wave patterns and their correlation with different states of consciousness, such as wakefulness, sleep, and various neurological conditions. Berger's invention provided a new way to study the electrical dynamics of the brain, paving the way for further exploration into neural oscillations and their role in cognitive processes and disorders. EEG has become an essential tool in both clinical diagnostics and neuroscience research, particularly in the study of epilepsy, sleep disorders, and brain-computer interfaces. Discovery of neurotransmitters, 1920s to 1950s. The discovery of neurotransmitters transformed our understanding of how neurons communicate. Otto Loewy's experiments in the 1920s demonstrated that nerve impulses are transmitted chemically between neurons. Loewy showed that stimulating the vagus nerve of a frog's heart released a substance, later identified as acetylcholine, that could slow the heartbeat of another frog, providing the first evidence of chemical neurotransmission. Subsequent research throughout the mid-20th century identified various neurotransmitters, such as dopamine, serotonin, and norepinephrine, and elucidated their roles in neural communication and behavior. Understanding neurotransmission has been critical for developing pharmacological treatments for psychiatric and neurological disorders and has informed the design of neurotechnologies that can modulate neurotransmitter activity to achieve therapeutic effects. Development of the first neural prosthesis, 1950s to 1960s. The creation of the first neural prostheses in the mid-20th century represented a significant step forward in restoring lost sensory and motor functions. Early cochlear implants, developed by researchers such as André Journo and Charles Uries, aimed to provide auditory sensations to individuals with severe hearing loss by directly stimulating the auditory nerve. These early devices demonstrated the feasibility of bypassing damaged sensory organs to restore function through direct neural stimulation. The success of cochlear implants has inspired the development of other neural prostheses, such as retinal implants for vision restoration and motor prostheses for individuals with limb amputations or spinal cord injuries. These advancements have shown the potential of neurotechnology to improve the quality of life for individuals with sensory and motor deficits. Introduction of Deep Brain Stimulation, DBS, 1987. Deep brain stimulation, DBS, emerged as a transformative treatment for movement disorders with the approval of its use for Parkinson's disease in 1987. DBS involves implanting electrodes in specific brain regions, such as the subthalamic nucleus or globus pallidus, to deliver electrical pulses that modulate abnormal neural activity. DBS has been shown to significantly alleviate symptoms in patients with Parkinson's disease, essential tremor, and dystonia, among other conditions. The success of DBS has led to its application in treating psychiatric disorders, such as obsessive-compulsive disorder and depression, highlighting its potential to modulate neural circuits and restore normal function. The development of DBS represents a critical milestone in the therapeutic use of electrical stimulation in neurotechnology. Emergence of functional magnetic resonance imaging, fMRI, 1990s. The introduction of functional magnetic resonance imaging, fMRI, in the 1990s provided a powerful, non-invasive tool for visualizing brain activity. fMRI measures changes in blood flow and oxygenation, which are correlated with neural activity, allowing researchers to map brain function in real time. fMRI has revolutionized the field of cognitive neuroscience by enabling detailed studies of brain function and connectivity. Researchers can now investigate the neural correlates of cognitive processes, emotions, and behaviors, leading to a deeper understanding of brain function and dysfunction. fMRI has also been instrumental in the development of brain-computer interfaces, BCIs, as it allows for the identification of brain regions involved in specific mental states and actions. Development of brain-computer interfaces, BCIs, 1990s present. The development of brain-computer interfaces, BCIs, in the 1990s marked a transformative moment in neurotechnology. BCIs enable direct communication between the brain and external devices, allowing users to control prosthetics, computers, and other technologies with their thoughts. This field has seen rapid advancements, with applications ranging from assistive technologies for individuals with disabilities to potential enhancements for healthy individuals. Early BCIs relied on EEG signals to detect brain activity associated with specific thoughts or intentions. Advances in neural decoding algorithms and machine learning have significantly improved the accuracy and reliability of BCIs, expanding their potential applications. Modern BCIs are being developed for a range of uses, including communication devices for individuals with locked-in syndrome, advanced prosthetic limbs that can be controlled with thought, and neurorehabilitation tools for stroke patients. Optogenetics, 2005. Optogenetics, developed by Carl Daceroth and colleagues in 2005, involves the use of light to control neurons that have been genetically modified to express light-sensitive ion channels. This technique allows for precise manipulation of neural circuits, enabling researchers to study the function of specific neurons and their role in behavior and cognition. Optogenetics has revolutionized neuroscience research by providing a powerful tool for dissecting the neural basis of behavior. Researchers can now activate or inhibit specific neurons with high temporal precision, leading to new insights into brain function and the development of potential therapeutic strategies for neurological and psychiatric disorders. The ability to precisely control neural activity with light has also inspired the development of new neurotechnologies for treating brain disorders. Advances in neural decoding, 2010s. Neural decoding, the process of interpreting brain activity patterns to infer mental states or intentions, has seen significant progress in the 2010s. Improved algorithms and machine learning techniques have enhanced the accuracy and reliability of BCIs. These advancements have led to more sophisticated applications, such as thought-to-text communication systems and more responsive neuroprosthetics. Modern neural decoding techniques leverage advanced computational methods to interpret complex patterns of brain activity recorded from multiple sources, such as EEG, fMRI, or intracortical electrodes. These techniques have enabled the development of BCIs that can translate neural signals into text or control robotic limbs with high precision. The progress in neural decoding has expanded the potential of BCIs to improve the quality of life for individuals with severe motor impairments and has opened new possibilities for augmenting human capabilities. Development of closed-loop systems, 2010s. Closed-loop systems, which can monitor brain activity and deliver real-time feedback, have become a major focus in neurotechnology. These systems hold promise for more effective treatments for neurological disorders by dynamically adjusting stimulation parameters based on ongoing neural activity. Examples include adaptive DBS for Parkinson's disease and responsive neurostimulation for epilepsy. Closed-loop neurostimulation systems continuously monitor brain activity and adjust stimulation in response to detected changes, providing a more personalized and effective treatment. For instance, adaptive DBS systems can modify stimulation parameters in real time to optimize symptom relief and reduce side effects. Similarly, responsive neurostimulation systems can detect the onset of epileptic seizures and deliver targeted stimulation to prevent them. The development of closed-loop systems represents a significant advancement in the precision and efficacy of neurostimulation therapies. Neuralink and brain-machine interface innovations, 2016. Elon Musk's Neuralink, founded in 2016, aims to develop advanced brain-machine interfaces to augment human capabilities and treat neurological conditions. Neuralink's ambitious projects include the creation of high-bandwidth, implantable devices that can seamlessly interface with the brain. This initiative has brought significant attention and investment to the field, accelerating technological advancements. Neuralink has focused on developing ultra-thin, flexible electrode arrays that can be implanted into the brain with minimal damage to surrounding tissue. These arrays are designed to record and stimulate neural activity with high precision, enabling more sophisticated BCIs. Neuralink's vision includes the potential for BCIs to enhance human cognition, memory, and sensory perception, as well as to provide new treatments for conditions like paralysis and brain injuries. Organoids and brain simulation, 2010s present. The development of brain organoids, miniature, simplified versions of the brain grown from stem cells, has opened new avenues for research. These organoids can model brain development and disease in vitro, providing insights into conditions like autism and Alzheimer's disease. Advances in brain simulation and modeling are also enhancing our understanding of neural processes and guiding the development of neurotechnologies. Brain organoids replicate key aspects of brain structure and function, allowing researchers to study the development of neural circuits and the effects of genetic and environmental factors on brain health. These models have been used to investigate the mechanisms of neurodevelopmentally and neurodegenerative disorders, screen potential drug treatments, and explore the effects of various interventions on brain function. The ability to simulate brain processes in vitro offers a powerful tool for advancing neurotechnology and developing new therapies. Expansion of non-invasive brain stimulation techniques, 2010s present. Non-invasive brain stimulation techniques, such as transcranial magnetic stimulation, TMS, and transcranial direct current stimulation, TDCS, have gained traction for both research and therapeutic applications. These methods can modulate brain activity without surgery, offering potential treatments for depression, anxiety, and other psychiatric and neurological conditions. TMS involves using magnetic fields to induce electrical currents in specific brain regions, modulating neural activity. TDCS uses low electrical currents applied through electrodes on the scalp to alter cortical excitability. These techniques have been shown to have therapeutic effects in conditions such as major depression, where TMS has been approved by regulatory agencies as a treatment. The expansion of non-invasive brain stimulation techniques represents a significant advancement in the accessibility and safety of neurostimulation therapies. Advances in neurofeedback, 2010s present. Neurofeedback, a technique that allows individuals to learn to control their own brain activity through real-time feedback, has seen significant advances. Modern neurofeedback systems utilize EEG, fMRI, or near-infrared spectroscopy, NIRS, to monitor brain activity and provide feedback, aiding in the treatment of conditions like ADHD, anxiety, and PTSD, and enhancing cognitive performance. Neurofeedback involves training individuals to alter their brain activity patterns by providing real-time feedback on their neural signals. For example, individuals with ADHD can learn to increase activity in brain regions associated with attention, leading to improvements in focus and behavior. Advances in neurofeedback technology have improved the accuracy and effectiveness of these interventions, expanding their potential applications and making them more widely available. Ethical and regulatory developments, 2010s present. As neurotechnology has advanced, ethical and regulatory considerations have become increasingly important. Issues such as privacy, consent, and the potential for cognitive enhancement raise complex questions. Regulatory bodies and ethical guidelines are evolving to address these challenges, ensuring that the development and application of neurotechnologies are conducted responsibly. Ethical concerns include the potential for misuse of neurotechnology, the implications of cognitive enhancement, and the protection of individuals' neural data. Regulatory frameworks are being developed to ensure the safety and efficacy of neurotechnologies, protect patient rights, and address ethical issues. The establishment of ethical guidelines and regulatory standards is crucial for the responsible development and deployment of neurotechnology. Brain-on-a-chip technology, 2020s. The development of brain-on-a-chip technology, which integrates neural cells with microfluidic devices, allows for the creation of sophisticated models of neural networks. These systems can mimic the architecture and function of the brain, providing a platform for drug testing, disease modeling, and the development of new neurotechnologies. Brain-on-a-chip devices combine neural cells with microfluidic channels to create complex, functional models of brain tissue. These systems can be used to study the effects of drugs on neural networks, investigate the mechanisms of brain diseases, and develop new treatments. The ability to create realistic models of brain function in vitro offers a powerful tool for advancing neurotechnology and improving our understanding of the brain. Integration of artificial intelligence, AI, 2020s. The integration of artificial intelligence, AI, with neurotechnology is driving significant advancements. AI algorithms can analyze vast amounts of neural data, enhancing the capabilities of BCIs, improving neural decoding accuracy, and aiding in the development of personalized treatment strategies for neurological and psychiatric disorders. AI techniques, such as machine learning and deep learning, can process complex neural data to identify patterns and make predictions. These capabilities are being used to improve the performance of BCIs, develop more effective neurostimulation therapies, and create personalized treatment plans for patients. The integration of AI with neurotechnology represents a major advancement in the field, offering new possibilities for understanding and treating brain disorders. Advancements in neural repair and regeneration, 2020s. Recent research has focused on neural repair and regeneration, aiming to restore lost functions after injury or disease. Techniques such as stem cell therapy, gene editing, and bioengineered scaffolds are being explored to promote neural regeneration and repair, offering hope for conditions like spinal cord injuries and neurodegenerative diseases. Stem cell therapy involves transplanting stem cells into damaged brain or spinal cord tissue to promote repair and regeneration. Gene editing techniques, such as CRISPR, can be used to correct genetic defects and promote neural recovery. Bioengineered scaffolds provide a supportive environment for neural growth and regeneration. These approaches represent promising strategies for restoring function after neural damage, advancing the field of neurotechnology and offering new hope for patients with neurological injuries and diseases.